Good afternoon. For our presentation, we wanted to do a study of the sickle cell disease. My name is Carlos Herrera, and this presentation was done with my partners, Faysal Ali and Guevarra. This presentation also served as part of our Biology 463 course that we are taking with Dr. Catherine Raftery, who also served as a mentor for the project. To begin, we wanted to give you guys a quick overlook of the sickle cell disease and how it manifests itself in individuals who suffer from it. Sickle cell disease patients have sickle-shaped cells that may accumulate in the bloodstream and cause a type of anemia, possibly leading to other health concerns that are much more severe. Patients also commonly suffer from periodic episodes of pain, which is caused by the vasoconstriction of the blood vessels in the body. Individuals are much more likely to be affected by it if they live in areas that are malaria-stricken. These include sub-Saharan Africa, India, Southeast Asia, and the Mediterranean areas of Europe. Individuals here in the United States are much more likely to have the disorder if they have African descent. The first description of the sickle cell disease was given to us in 1910 by Dr. James B. Herrick, who took a blood sample from a dental student of African descent. What he found in that blood sample were thin, elongated, sickle and crescent-shaped cells. Inadvertently using the name sickle shape led us to the disease's eventual naming later on. In 1915, Dr. Jerome Cook and Dr. Jerome Meyer described another early case of the sickle cell disease and related it to Dr. James B. Herrick's 1910 paper. They also introduced the idea that the disease may be inherited, commenting that the patient's siblings also suffer from anemia. In 1933, Dix, Amand, and Bibb demonstrated that there were differences between the sickle cell anemia and the sickle cell trait. However, the genetic differences were still unknown. In 1949, University of Michigan researcher James B. Neal published an article in which he demonstrated that the inheritance pattern of the disorder was autosomal recessive, meaning that an individual must inherit two copies of a mutated allele from both parents in order for the disorder to be manifested by the individual. Neal came to this conclusion by utilizing the data obtained from individuals of African descent. That same year, Linus Pauling and his colleagues conducted experimental analysis on the hemoglobin found in red blood cells. The experiments were conducted using gel electrophoresis using blood samples from both normal and affected individuals. The results of these experiments showed abnormalities in the shape of the hemoglobin of affected individuals, which led researchers to name sickle cell disease as a molecular disease, the first of its kind. In 1953, South African geneticist Anthony Allison conducted a study of the Luo tribe people who live in areas close to Lake Victoria, an endemic malaria region. The results of the study were published in 1954 and found that the heterozygote individuals developed a much milder infection due to lower P. falciparum, the bacteria that causes malaria, counts being found in their blood. The results of this led to the hypothesis that the sickle cell trait provides a heterozygote advantage in increasing fitness against malarial infections. In 1957, using gel electrophoresis and chromatography, Ingram and Stratton discovered that the sickle cell hemoglobin was caused by a single amino acid base substitution at the sixth position of the beta globin chain. The substitution resulted in glutamic acid being substituted for valine, therefore altering the protein structure of beta globin chains. Goldstein and his colleagues in 1963 went a step further by conducting a multitude of analyses on the tryptic peptide chains of affected and normal individuals. The results of this analysis led to the conclusion that the amino acid substitution affecting sickle-shaped hemoglobin was caused by a missense mutation at the codon number 6. This base substitution change was an adenine being changed into a thymine, changing the amino acid being synthesized in the protein structure of beta globin. In 1984, we had the first documented case of bone marrow transplant being conducted on a patient. This transplant sought to create normal red blood cells from the bone marrow being implanted into the individual. In 1998, the FDA approved hydroxyurea, the first medication that treats against sickle cell disease. Hydroxyurea makes the red blood cells bigger, it makes them stay rounder and more flexible, and it makes them less likely to turn into a sickle shape, which is what is found in individuals who suffer from sickle cell disease. In 2016, the first instance of stem cells being transplanted into an unrelated donor from a children with sickle cell disease was noted. The issue found before was that individuals who needed to receive a bone marrow transplant needed to be related or needed to be a match, which is very difficult to find. In 2017, the first reported use of gene therapy technology to treat sickle cell disease was given. However, this gene therapy technology is still in its very early stages in helping us treat against sickle cell disease. The current treatments and cures for sickle cell disease are blood transfusions that are done in order to manage the acute cases. Hydroxyurea may also lead to increased expression of fetal hemoglobin, which helps in the offset the impacts of sickle cell disease. Bone marrow transplants are the only known cure for sickle cell disease. However, as I mentioned, finding donors is very difficult due to the polymorphic nature of HLAs and how difficult it is to find matching individuals. What we have for the future of sickle cell disease are very several paths. Some of these includes stem cell transplantation using modified or donor healthy stem cells from our own person or from other individuals in order to replace the mutant cells found in sickle cell disease. Gene therapy, as I mentioned, is also in its early stages, but it may help drive the expression of fetal hemoglobin in order to fix the disease causing mutation entirely. Thank you guys for your time. Hopefully, you guys enjoy this presentation. And if you guys have any questions, make sure to leave it in the comments. Thank you very much, and have a nice day.